The overall goal of this procedure is to produce a quantifiable, reliable, and reproducible preclinical animal model of respiratory and non-respiratory insufficiency induced by a cervical spinal cord injury. This is accomplished by first preparing the instruments and the drugs required for the procedure. The second step is to anesthetize the animal, then intubate it and connect it to a rodent ventilator. Next, the spinal cord is exposed and the cervical spinal injury is induced. After sewing up the animal, the anesthesia is removed and appropriate post-surgical care is provided. Ultimately, histological techniques, as well as nerve and muscle recordings, are used to examine the extent and effects of the cervical injury.
The main advantage of this technique over existing methods, such as spinal cord contusion, is that the injury is smaller and well-defined. Demonstrating the procedure will be Emilie Keomani, an engineer from my laboratory.
In preparation for surgery, clean and autoclave the surgical instruments. Also prepare a 2 by 1 milliliter syringe with the pre-anesthetic drugs and complete the volume to 1 milliliter with lactated ringers. In another syringe, prepare the reversal for the pre-anesthetic drugs. After subcutaneously administering